Hi guys, this is Dr. Hamid, an ECVMG certified physician, and we are continuing our high yield revision for the microbiology for the USMLE exam. So this is our second episode here. So we've got anaerobes versus facultative anaerobes. So the true anaerobes lack catalase and superoxide dismutase. So these enzymes are very high yield for the bacteria to neutralize toxic oxygen radicals. So the bacteria use catalase normally and superoxide dismutase to neutralize these toxins, and anaerobes lack these enzymes, so they are susceptible to oxidative damage. So they are normal GI flora, but they are pathogenic in low oxygen environment. This is why the anaerobes are involved in intra-abdominal infections or in ischemic tissues, for example, which are very far away from oxygen, and they are rare in upper respiratory infections. So this is for anaerobes versus the facultative anaerobes, which entails some of it includes some of the most famous organisms such as strep, staph, and intergram negative. So facultative anaerobes, they can live near the oxygen and also behave like anaerobes away from oxygen. For example, this is why staphylococcus, for example, can cause upper respiratory tract infection, but can also be involved in abscesses and in infection in anaerobic and ischemic tissue. They can use oxygen as a terminal electron acceptor, or use fermentation, so they can get energy both through aerobic and anaerobic way. This is obligate anaerobe versus the facultative anaerobes. And we've also got obligate intracellular organisms. These organisms rely on the host ATP to get energy. They can't be independent out of it, and this gives them major virulence because they are staying away from antibodies, so the antibodies can't fight them. And as we've discussed in our last episode, these organisms can't be stained, for example, by gram stain because they are intracellular. This includes rickettsia, chlamydia, and Coxiella. So you have to know that Coxiella, chlamydia, and rickettsia, they are obligate intracellular, so you have to find them inside of cells, not outside of cells, versus the facultative intracellular. So these organisms can be inside and outside of cells, such as Salmonella, Neisseria, and the Mycobacterium. So we've got some pretty unique organisms here. So for example, the Mycobacteria live inside of the macrophages, which we're supposed to fight these bacteria, but they can live inside of the macrophages and evade the phagolysosomes. And this is why we get granuloma, as the body tries to limit this infection by forming a granuloma of these macrophages around them, and the Salmonella infectus bears patches in the GI tract and may use the macrophages as a vehicle to transfer across the body. So these are facultative intracellular organisms, Salmonella, Neisseria, and the Mycobacteria, versus the obligate intracellular such as Rickettsia, Chlamydia, and Coxiella. And we are going to talk about another virulence factor, which is the capsules. It's a very important. So we have to know that the strep pneumonia, Haemophilus influenza, and the Neisseria meningitis are very high yield because we give vaccination for the people who have had splenectomy, or for example to sickle cell disease young patient who have splenic infarctions normally associated with sickle cell disease. So these people get the vaccine for strep pneumonia and Haemophilus influenza and Neisseria meningitis because these are encapsulated bacteria and they are opsonized, and the phagocytosis happens through macrophages which are present in the spleen. So if we miss the spleen, we are endangered by these organisms. The another organisms is Pseudomonas aeruginosa, which we are going to discuss in detail later on, and also E. coli, Salmonella, and Klebsiella, and also group beta strep. So the strep pneumonia, Haemophilus influenza, and Neisseria meningitis, and also other organisms such as Pseudomonas, E. coli, and Salmonella. So this is a virulence factor. As we've discussed also, that the virulence factor is something that either helps the bacteria invade or evade like the immune system, and the polysaccharide capsule serves as antiphagocytic shield. It like covers and hides the succeeding layers of the bacteria and from the immune cells. And we may use this like a capsule as a and can be conjugated to a carrier protein and the vaccine such as the strep pneumonia vaccine. Next, we are going to take about the splenic patient, as we've discussed in a previous slide. So these bacteria, these encapsulated bacteria, are opsonized. So the opsonization means that they are marked. So they are marked by antibodies and the complement system. And after being marked, when they pass through the spleen, the splenic macrophages are the primary site for clearing these opsonized pathogens, and we've got a major problem in post-splenectomy or, for example, sickle cell disease, and these are Neisseria meningitis again, strep pneumonia, and Haemophilus influenza. And we are going to talk about the urease positive organisms. So you have to know that urease is an enzyme that acts on the urea to hydrolyze it into ammonia and carbon dioxide. So you know, like the ammonia is a basic, it's basic, and this serves some organisms a favor, and by favoring their growth. For example, the H. pylori. So the H. pylori lives in the acidity of the stomach, and being a urease positive organism can help this bacteria thrive in the acidic environment in the stomach. And also the Proteus and the Staph saprophyticus can cause a urinary tract infection, and they also urease positive, they increase the pH, and they predispose to stereovite stones. So the stereovite stones are called magnesium ammonium phosphate. So they are for like part of their ingredients are like the ammonia. So part of their ingredients are the ammonia. So these are the stereovite stones formed by the Proteus and the saprophyticus, and the Proteus is very high yield because it is associated with a unique type of stone called the staghorn stone, which is a very big type of stone because it has potent urease activity, and another less high yield or urease positive organisms such as Klebsiella and the epidermidis, for example, Nocardia, Cryptococcus, and Uriplasma. So this is about urease positive organisms. We've got also catalase positive organisms, and their clinical importance in a disease called the chronic granulomatous disease. So you have to know that the catalase is an enzyme that the bacteria may have or may not have that degrades H2O2, which is toxic to the bacteria, into some neutral substances such as water and oxygen bubbles. So they turn this very like dangerous molecule, which is H2O2, into water and the oxygen. Some of the people have a disease called the chronic granulomatous disease. So these patients have NADPH oxidase deficiency, and the NADPH oxidase is supposed to provide like to provide H2O2 to fight these bacteria. So these patients have a limited supply of H2O2, and this catalase positive organism will like degrade this limited supply of H2O2. So these patients who are having like chronic granulomatous disease have so little amount of H2O2 because they lack the organism, they lack the enzymes that produces H2O2. So the catalase positive organisms are pretty dangerous here, such as Staphylococci, E. coli, and H. pylori. So you may find in a vignette of a question people who are having chronic granulomatous disease, having recurrent infection with staphylococci, E. coli, or H. pylori, because these organisms are catalase positive. These people have a limited H2O2 because they have like NADPH oxidase deficiency. So this is about catalase positive organisms and their association with chronic granulomatous disease. And we are going to talk about pigment producing bacteria. So you have to memorize this stuff. So Actinomyces israeli produces yellow or sulfur granules, which are composed of like the filaments of the bacteria. So Actinomyces israeli with the yellow sulfur granules, and Staphylococcus aureus is associated with golden yellow pigment. So you know, like aureus equals gold, so Staphylococcus aureus, as it appears here, it is associated with production of a golden yellow pigment. So yellow sulfur granules with Actinomyces israeli, and golden yellow pigment with Staphylococcus aureus. Pseudomonas aeruginosa with the production of blue-green pigment, which are biocyanin and pyoverdin. So you know, like some like wounds have like some greenish to bluish color is due to biocyanin and pyoverdin when they are infected with Pseudomonas aeruginosa. And the Serratia marcescens is associated with the production of a characteristic red pigment. So red pigment with Serratia marcescens, blue-green pigment, biocyanin and pyoverdin with the Pseudomonas aeruginosa, golden yellow pigment with the Staph aureus, and yellow sulfur granules with Actinomyces israeli. And there's another concept which is biofilm producing bacteria. We've discussed this in the previous episode also. So this biofilm helps the bacteria and is a virulence factor by helping them stick to devices such as urinary catheters and also central venous catheters. So we are going to discuss these bacteria. First is a Staph epidermidis. So this bacteria adheres to catheters and prosthetic devices via biofilm formation. So this is the Staph epidermidis, and how the biofilm is a virulence factor for it by helping the bacteria adhere to catheters and prosthetic devices. And the Verdens streptococci, so the Verdens streptococci produces dental plaques, and this is why we give antibiotics before dental procedures, because they can adhere to heart valves and cause infective endocarditis. So this is another application for the biofilm like production by the bacteria, first in the Staph epidermidis in catheters and the prosthetic devices, and the Verdens streptococci with the production of dental plaques and the ability to stick to the heart valves and cause infective endocarditis. Also we have Pseudomonas aeruginosa. They use the biofilm to colonize the respiratory tree, and they are like important organism, and it is highly tested in the vignettes in cystic fibrosis. So the association of Pseudomonas aeruginosa with upper respiratory tract infection in cystic fibrosis is very important, and the Pseudomonas aeruginosa is also as a major cause of ventilator associated pneumonia. So it's very high in yield to memorize this. Non-typable Haemophilus influenza with the otitis media through biofilm, like also resistance. So all of these bacteria use the biofilm to either stick to the devices, dental plaques, or to heart valves. And the cystic fibrosis patients having upper respiratory tract infection, because the Pseudomonas aeruginosa use this biofilm like ability to colonize the respiratory tract. So that's very high yield stuff. We are going to talk about the spore. So the concept of a spore is the way of the bacteria to protect itself when the environment is harsh. For example, in like a harsh environment, and it is a major cause of the persistence of this bacteria in the ICU and in the hospital, and causing hospital like acquired infection. The most important thing to know is that the spores are pretty unique to gram-positive. The gram-negative cannot form spores. So this is the first point you have to know. And they enter a dormant states when the nutrients are limited, forming these highly resistant spores. So this is the spores contains the genetic material. So it's like main purpose, it is tries to protect the genetic material of the bacteria until the environment is favorable again, and it contains something called the dipicolinic acid. So it's very high yield to know that the dipicolinic acid, because it helps these spores to resist heat and chemical and radiation, and these spores lack metabolic activity. So this is assisted by dipicolinic acid. And our way to eliminate these spores is either autoclaving in 121 Celsius for 15 minutes, or using iodine-based sporicidal agent. So you either use iodine-based sporicidal agents or autoclaving at 121 for 15 minutes to kill the spores. So these spores have dipicolinic acid to stabilize it, and it have genetic material at the core. It has cortex and have thick peptidoglycan layer. Remember from our previous episode we've talked about that the gram-positive have thick peptidoglycan layer, where the gram-negative have thin peptidoglycan layer. We've got two major families here, which are the Bacillus anthrax and the Clostridia. So the Bacillus anthrax, Bacillus cereus, for example, so under the Bacillus, and the Clostridium with either tetani or Bifrenigens botulinum. So it is either Clostridium and or either Bacillus, which are cereus or anthrax, for example. So these are our examples, our primary examples for the spore forming bacteria. And again, we've talked about the capsular polysaccharide and how it is antiphagocytic, virulence factor. It prevents phagocytosis and inhibit complement-mediated lysis by masking the bacterial surface antigen. And you have to know that it is highly charged and hydrophilic structure, and it acts as a physical barrier to a host defense. We've talked about that already. We've got some another virulence factor, which is protein A. So the protein A is a key virulence factor, and it is specific to Staph aureus. So what does protein A do? It halts the immune system by binding to the Fc region of IgG and preventing opsonization. We've talked that the opsonization is how the bacteria is marked to be phagocytosed later in the spleen, for example. So the protein A of the Staph aureus, so the Staph aureus have got this protein A, and the protein A binds to the Fc region of the IgG. So this is the antibody have a certain 3D structure, and by the Staph aureus protein A binding to this Fc region, it flips this antibody. So it makes another configuration and interrupts the opsonization process. So the protein A interrupts the opsonization process by binding to the Fc region of the IgG and interrupting the 3D structure by flipping the antibody. This is how it is a virulence factor for Staph aureus. We've got also IgA protease. The IgA is a surface antibody. It's pretty important in the respiratory tract and genitourinary tract, specifically in the respiratory tract. And some bacteria have got something to break it out, so can break this IgA. It's called the IgA protease. So this enzyme cleaves the secretory IgA, allowing the bacteria to adhere and colonize mucous membrane. Also, we've got M protein. So the M proteins is pretty famous, and it's highly tested. So it is expressed by group A streptococci. Group A streptococci is associated with rheumatic fever, but the main purpose for the M protein for these bacteria is not like the rheumatic fever, but its main purpose is the prevention of phagocytosis, and rheumatic fever is a collateral damage, so we're going to explain. So the M protein, so again, its main purpose is the prevention of phagocytosis. So the bacteria have this M protein to protect itself from being phagocytosed, but as a collateral damage, this M protein shares some sequence homology with the human cardiac myosin. So the body, like immune systems, forms like antibodies against this M protein and against this bacteria, but it shares the sequence homology with the human cardiac myosin, so the immune systems attacks our human cardiac myosin, and unfortunately we have acute rheumatic fever as a collateral damage by being infected with the group A streptococci. We are going to talk about how the bacteria like may have sex. So like the bacteria can have sex, so which is through different like mechanisms. First, we are going to talk about transformation. Transformation is not a sexual process, but it involves the uptake of a naked environmental DNA. So we've got bacteria here in this figure, and we've got another bacteria that is dead, and its genetic material is fragmented, and it is there in the environment. So the live bacterial cells takes these fragments of a dead bacteria and incorporate it. So the competent bacteria or live bacteria binds and import these short pieces of the naked environmental DNA from some lysed cells. And we are proving that by adding something called the deoxyribonuclease or DNA. So if we added deoxyribonuclease or a DNA, is going to break this free DNA fragments, and the transformation is going to be halted, and this proves the process of transformation. So this is the experience experiment of adding deoxyribonuclease. And there are some common organisms such as streptopneumonia, Haemophilus influenza type B, and Neisseria. These Shin organisms are some of our examples of like applying these methods of acquiring genetic material. And one very important thing you have to notice here, that transformation doesn't require the direct cell-to-cell contact, because it happens with a competent cell or live cell and a dead cell. So it does not involve this direct cell-to-cell contact. You have to know this is called transformation. This is called again transformation. And the other example, which is sexual contact in the bacteria, is conjugation. So we have F positive and F negative bacteria. What we mean by F positive, if they have plasmid. If the bacteria have plasmid that encodes for the sex pilus, it is called F positive, aka male bacteria, versus F negative, which are the female bacteria. So the F positive, F negative conjugation is a process that transfers plasmid DNA via sex pilus. So we've got here F positive bacteria. It has this plasmid. So this plasmid is what encodes the sex pilus here. So the sex pilus here is what called what forms the bridge between the two bacteria, and this is the recipient cells, which is F negative. So this requires direct cell-to-cell contact through sex pilus encoded by the F positive plasmid, and a single strand of plasmid DNA is transferred. So we've got here a single strand of plasmid DNA is transferred. You have to notice how the main DNA chromosome stays the same. There is no exchange between the F positive and F negative bacteria of the main genetic material. It's only one strand of this plasmid, which is transferred between the two bacteria, and following the transfer, the recipient synthesizes the complementary strand. So here the recipient synthesizes another complementary strand. So you have noticed how the recipient one, which was a female, which was F negative, is now transformed also to being F positive. So as a result of this F positive F negative conjugation is that we have both of them are males now, both of them are F positive bacteria. So there is no chromosomal DNA. Again, no chromosomal DNA is transferred in this specific process, so the chromosomal DNA stays the same. So this is F positive, F negative conjugation. And there's another type of conjugation which is called high frequency F negative conjugation. So but what we mean by high frequency recombination is that if positive plasmid incorporates into the bacterial chromosome and the transfer is between the leading part of the plasmid and the flanking chromosome. So we're going to break this. And so we've got here F positive cells and F negative cells as our previous example, but this plasmid is incorporated here in the chromosomal and the main genetic material. And also we've got six pillas and the bridge, like what happened in our previous F positive F negative conjugation. But the first difference here is that we've got the plasmid, and which is going to be transferred, but it is going to be transferred along with flanking bacterial chromosome. So this is the main difference here. There is flanking chromosomal genes that are transferred with the plasmid, and the conjugal bridge often breaks before full transfer, so the recipient usually remains F negative. So high frequency recombination or conjugation transfers some chromosomal material, while our previous examples there is no transfer of chromosomal material, but at the same time the daughter bacteria here remains F negative. So our previous example transferred the daughter bacteria to a male, but here not. So these are the two main difference between them, and the recipient acquires new bacterial genes that can be integrated into its own gene. So high frequency recombinations entails the exchange of chromosomal genes, but at the same time often usually the F negative bacteria remains F negative after high frequency conjugation. Next, we've got another method by which the exchange of genetic materials happens is through the phage. So the phage is have through multiple cycles. First, there is a lytic infection, so the bacteriophage here infects the bacterium, and it goes on and inserts its genetic material, and eventually there is lysis of the genetic material here, and this genetic material is during something called the packaging error. So that during the phage assembly, so it is supposed to take its own genetic material that he injected into the bacteria, but by error, as a packaging error, during phage assembly, bacterial DNA fragments are accidentally packaged into the phage capsid instead of viral DNA. So we've got some phage that has taken its own genetic material, and others that has taken the genetic material by blue here of the bacteria. So this is a packaging error. So that phage is defective and is going to infect another bacterium and is going to inject the DNA or the genetic material of the previously infected bacteria. So this is called the generalized transduction, because any part can be part of this packaging error. So this packaging error is random, and any part can be inserted into this phage, and this phage is going to infect another bacteria, and is going to transfer it to another bacterium, and this is called the generalized transduction. And there is another thing is called specialized transduction, and it depends also on the phage, and occurs during the lysogenic stage. So the phage may be like genetic material may be incorporated here into the genetic material of the bacterium. This is called lysogenic cycle. So the prophage DNA is incorrectly excised from the bacterial chromosome, so it takes some piece of like the genetic material of the bacterium, and excision error causes flanking bacterial genes to be packaged in the new phage capsid, and this is a key driver of virulence evolution and the transfer of specific genes other than random like errors that occurs with the generalized transduction. You have to memorize some of these like virulence if like factors that are essentially toxins. So group A strep erythrogenic toxin happened like this way: botulinum toxin, cholera toxin, diphtheria, and the Shiga toxin. So group A strep, botulinum, cholera, diphtheria, and the Shiga. So the diphtheria and the Shiga toxin, cholera and the botulinum, and the group A strep erythrogenic toxin happened this way through something called a specialized transduction, where there is excision error and the phage genes are like not taken only, but there is a flanking part of the bacterial genes that are being excised incorrectly with these phage genes, and this is a way of virulence evolution for specific toxins such as group A strep erythrogenic toxin, botulinum, cholera, diphtheria, and the Shiga toxin. And there lastly we've got something called the transposition. So the transposition facilitates the spread of multi-drug resistance. So the transposons are like jumping genes. So we've got like in a previous example, there is a DNA of the bacteria and there is a plasmid. So the transposons are like jumping genes, which are DNA segments that can move within like or between the DNA molecules, so they can copy or excise themselves into the same or a different DNA, like from plasmid to the chromosome. So they can either go in the different parts of the chromosome or from plasmid, for example, to chromosomes. These genes are called the transposons. So this process is critical for rapid accumulation of multiple resistance genes on a single plasmid. And as a clinical example is the transfer of VAN-A gene. So the transfer of VAN-A gene from Enterococcus to Staph aureus is what how we've got vancomycin resistance Staph aureus. So we've got vancomycin resistant. You have to memorize this example because it is an example of how transposition facilitated multi-drug resistance, which is the transfer of a VAN-A gene from Enterococcus to Staphylococcus, and hence the Staph aureus is now vancomycin resistant Staph aureus, very high yield. And lastly, we are going to talk about the difference of exotoxins and endotoxins. So the endotoxins are part of the bacteria, while exotoxin is something that the bacteria has produced. So this is main difference. Endotoxin is part of the bacteria, but it is part of only gram-negative. The gram-positive can't have endotoxin, and it's essentially the lipid A. Remember from our like last episode, we've talked that the gram-negative has outer membrane, while the gram-positive don't does not have a outer membrane. So the lipid A component of lipopolysaccharide of the outer membrane of gram-negative is essentially the endotoxin, and by being a part of the bacteria, it have low toxicity. So it have low toxicity. We have like accumulation of so many bacteria to got like high toxicity. So the fatal dose in hundreds of micrograms. And the clinical reaction of it is non-specific, pretty non-specific, which is called fever, shock, and the DIC. So we've got a reaction from the body, which is fever, shock, and the DIC to this parts of the bacteria, is essentially the lipid A of lipopolysaccharide. and this is stable at 100 Celsius for 1 hour, and there's essentially no vaccine because it is part of the bacteria, no toxoid, no vaccines available. On the other hand, the exotoxin is something that is produced by the bacteria, so it is not, it can be in gram positive, in gram negative, not only in gram negative like this lipid A component that is part of the outer membrane in gram negative. And the chemistry, it is, for the exotoxin, is a polypeptide, and it has high toxicity, so think of it like a venom of a snake, so it is highly toxic in fatal doses of 1 microgram. So it's kind of specific, for example, Shiga toxin, tetanus toxin. So all of these toxins that we are going to study are like exotoxins, and they destroyed at 60 Celsius, like versus the endotoxin, which is more stable and destroyed at 100 Celsius. So we've got vaccines for these toxins, which are called the toxoids. So we've got toxins and the toxoids. So the toxoids are vaccines here. I hope you guys enjoyed our video today, and see you next videos of our very high yield revision for the microbiology. Thank you.